This lecture is going to examine different ways in which we can target angiogenesis. There's two main drug groups or drug classes, the monoclonal antibodies and the small molecule tyrosine kinase inhibitors. Probably the prototype in terms of targeted therapies, the first one we usually think of is bevacizumab, and this is under the brand name Avastin. Typically, we don't refer to brand name drugs, but I do it for this particular one because it is clinically relevant. And if you look in the literature, these targeted therapies are made so quickly and go through so many clinical evaluations so rapidly that there typically is no generic available. So it's really only one or maybe two manufacturers that will make these drugs. So bevacizumab is a monoclonal antibody. Typically we refer to these as MABs for the shorthand of that. And they bind to VEGF. And so this is going to be soluble VEGF that's kind of floating around in the bloodstream. And of course that stands for vascular endothelial growth factor. And it's really a major driver of the process of angiogenesis. And so obviously if you inhibit that signaling component of angiogenesis, you're going to severely restrict the ability of a lot of these solid tumors to grow and proliferate. And so clinically, we see this drug used as an adjuvant therapy in several tumor types, ovarian, lung, colorectal, brain, renal cancer, as well as cervical cancer, and others. And what's important to remember is that the dosing regimen is unique, and the way in which this drug appears in these regimens and the frequency of it is going to be unique depending on the tumor as well as the tumor subtype. And so that's kind of beyond the scope of this course. That's more for the guys and gals in oncology fellowships. So just in general, when we talk about monoclonal antibodies, there's two main things to kind of realize or understand. One is the specific receptor or protein that the monoclonal antibody is targeted to. And then two is the overall mechanism of how that works. And so it doesn't really matter which monoclonal antibody you're talking about. Anything that ends in MAB has pretty much the same mechanism of action. The only thing that's different is what it's going to bind to. Okay, so there's three main mechanisms of action when we talk about these particular drugs. And the first is that we can see direct killing of the target cell through two more or less immune-mediated processes. And those processes we've probably talked about at some capacity over the part of this curriculum, but that is antibody-dependent cell toxicity, or ADCC, and complement-dependent cytotoxicity, just often called CDC. And so CDC typically involves fixation of the complement cascade to the immunoglobulin Fc region. And of course, that's going to obviously activate the complement cascade. And there will be an enzymatic piercing of the tumor cell, and then obviously tumor cell death. Antibody-dependent cell toxicity, or ADCC, is really most of where oncology monoclonal antibodies kind of reside. We think of effector cells such as monocytes, macrophages, and granulocytes, and some other types of lymphocytes that express a lot of these receptors that bind to the Fc portion of a immunoglobulin molecule. And then you get to see these effector sites binding to the antibodies, and they adhere to the tumor cells, and they subsequently mark the tumor cell for lysis and destruction. And so the Fc region has to be present for either of these two processes to occur, and so typically only intact antibody molecules will be effective for this type of strategies. Okay, so the one is, so the first mechanism is really direct killing through either of those two processes. The second mechanism is we can combine a monoclonal antibody to a conjugated toxin, and then this conjugated toxin will move through the plasma membrane by way of its binding to this receptor, and then it will be internalized. And that that toxin can be a radiologic isotope. It could just be a conjugated toxin that is from the environment. Several different toxins have been developed, and these can be applied to these monoclonal antibodies, and they can offer very specific, very targeted delivery to the cancer cell. And then third, and this is probably the most kind of foundational, is that the monoclonal antibody basically just binds to whatever molecular target it was developed to, and essentially shuts down that cell process. And so if that cell process is involved with cell growth, proliferation, angiogenesis, etc., it will stop that process and then contribute to the cancer cell's demise. And so these are three different mechanisms, all of which are depicted here. A naked monoclonal antibody is really just something like bevacizumab, where it would cause tumor cell death either through those two processes we just talked about, ADCC or CDC. And then there's other ways in which we can cause tumor cell death by conjugating that monoclonal antibody to a toxin. That can be an immune toxin. It can be a small drug that becomes activated upon binding. It can be a liposome. It can be bound to other immune cells or trafficked to other immune cells. So there's different ways in which this can occur, but these are the two probably main ones that are used for cancer chemotherapy. When we talk about inhibiting angiogenesis, and we talk about just in general VEGF inhibitors, we're talking about a very large family of drugs and receptors that all basically lead into the process of angiogenesis. They can also lead into the ability of tumor cells to migrate, proliferate, survive, and then also become permeable. So, kind of going along with migration, kind of moving from one compartment to the next. And these are, of course, the hallmarks of cancer, first described by Hanahan and Weinberg. But you can see a lot of these different receptors exist. There's at least 3 VEGF receptors, and there's a few drugs that bind not only to the receptor, which is shown here for ramucirumab, but also to the ligand, which is soluble VEGF, which is, there's obviously different isoforms of that. So bevacizumab binds to the ligand, and some of these other drugs bind to the receptor. Okay, both of these are monoclonal antibody, and you can tell that by the name. The suffix mab, M-A-B, is short for monoclonal antibody. There's other monoclonal antibodies in development, in different phases of development. Some of these have been eliminated over the years, but the overall general concept of this diagram is correct. And then, of course, the next group of drugs, which we'll be talking about shortly, are the small molecule tyrosine kinase inhibitors, and these all have the suffix NIB, or NIB, at the end of them, and we'll focus on 2 very specific NIBs that play into the VEGF receptor signaling cascade, which is shown here. So here are the current FDA-approved drugs that are classified as angiogenic inhibitors, and they all inhibit some component of this cascade, which is again kind of shown here on the right. Here you've got bevacizumab inhibiting vascular endothelial growth factor, the ligand. There are other drugs, cetuximab and pantizumab, that inhibit epidermal growth factor receptor, its cousin. I do want to point out that VEGF and EGFR, both of these receptors are tyrosine kinase inhibitors, so there is some overlap. And then there also is some overlap on the intracellular portion of these tumor cells in terms of how these tyrosine kinases communicate with the outside world. And so they take signals from the outside and then they internalize them, and that activates these protein cascades. And if you've looked at some of the cytogenetics from either pathology or genetics, you might be familiar with the RAS, RAF, ERK, or mTOR pathways, and how all of these altered genetic mutations can lead to cell growth, proliferation, and survival of the tumor cells. Understand too that all of these drugs, some of them are monoclonal antibodies such as bevacizumab, some of them are small molecule kinase inhibitors, ones that end in NIB. Everolimus is on this list. It's classically an immunosuppressant suppression type drug, but it affects negatively the mTOR pathway, so it's just a downstream effect, and so that will indirectly inhibit angiogenesis. And then there's a whole bunch of other small molecule tyrosine kinase inhibitors listed here, and we're only going to focus on a handful just to make it a little bit simple to digest. And then this diagram also kind of reiterates the major point is that we've got drugs that inhibit the ligand, we've got drugs that inhibit the receptor, and we've got drugs that inhibit the internal cascade of what this specific receptor does on the inside of the cell. So there's a lot of chemistry and a lot of molecular biology in terms of the development of these drugs, and we'll leave off here and we will continue with the tyrosine kinase inhibitors, which are the next group of drugs.